Hello students, welcome to EPG Pathshala. I am Professor Pankaj Madan from Faculty of Management Studies, Gurukul Kangri Vishwavidyalaya, Haridwar. Today we are going to talk on module Analysis of Variance and Experimental Design conclusion under the paper Quantitative Techniques for Management Decisions. After the completion of this module, the students, you will understand why different designs of experiments are required, what is the general purpose ANOVA, how the ANOVA test is conducted, and also in the table you will see how the one-way ANOVA is done. You will see a summary table of a two-way ANOVA. You will also see a summary table of ANOVA of a complete randomized design and a randomized block design. Why different designs of experiments are required? If we want to set an experiment or you want to test an hypothesis, and if you want to check which shift is more productive, or if you want to check the difference in the production levels of various machines in a manufacturing organization which is running production of goods in 3 shifts, then we need to design this experiment or we need to design this test. Each problem may have a different design. In this problem, we are talking about a problem of a manufacturing unit or a manufacturing company running production in 3 shifts. A design for an experiment defines the size and number of experimental units. It also tells the manner in which the treatments are allotted to the units, and also the appropriate type of grouping of testing units, that which unit will be in which group. Experiment or a test actually starts with a problem and an answer to which is obtained from interpretation of a set of observations collected suitably. For this purpose, a set of experimental units and adequate experimental material are required. Each size of plot of land, a single or a group of plants are used for agriculture experiments. For industrial experiments or industrial tests, machine, man, method, material, they form the experimental unit. For a social test or a social experiment, product, services, persons form the experimental unit. Requirement of various types of designs ensure validity, interpretability and accuracy of the results obtainable from the analysis of these observations. Now, you would be interested to know that what are the objectives which are served by these principles. So, these are the randomization which defines the manner of allocation of the treatments. What does it mean? It means that randomization tells us the way of arrangement of these allocation units. The replication specifies the number of units to be provided for each of the treatments, or you can say it is the number of units in each arrangement. Suppose we are talking about a set of machines in a shift. Then in one shift, if we are talking about different machines, then how many machines will be specified in one shift? It will tell about that, and also in each machine how much production is being done, that is also set by these principles. The third is error control. Error control is one which increases the precision by choosing the appropriate type of experimental units and also their grouping. For these different arrangements of population or samples, various experiments or testing techniques are used. And what are those? You already know: one-way ANOVA, a two-way ANOVA, or MANOVA. Statistical model and analysis of variance. A statistical model is actually a linear relation of the effects of the different levels of a number of factors involved in an experiment, along with one or more terms representing error effects. The effects of any factor can be either fixed or random. For example, the effects of two well-defined levels of irrigation are fixed, as each irrigation level can be reasonably taken to have a fixed effect. Again, if the variety of a crop is taken as a factor with the number of varieties of crop as its levels, then the effects of the varieties will be random if these varieties are selected at random from a large number of varieties. A random effects can again belong to a finite or a infinite population. The error effects are always random and make either to a finite or infinite population. A model in which each of the factors have fixed effects and only the error factor is random is called a fixed model. Models in which some factors have fixed effects and some random effects are called mixed models. Again, models where all the factors have random effects are called random models. So depending on the fitness or otherwise, the random effect populations, mixed and random effect models can be of many different types. In fixed effect models, the objects are to estimate the effects, find a measure of variability among the effects of each of the factors, and finally find the variability among the effects of each of the factors, and finally find the variability among the error effects. In random effects models, the main emphasis is on estimating the variability among the effects of the different factors. The methodology for obtaining expressions of variability is, however, mostly the same in the different models, though the method appropriate for testing are different. A fixed effect model for, say, two factors are, they can be written as you can see into this formula: yijk plus mu ai plus bj plus eijk, where yijk is an observation coming from a unit defined by the levels. Ijk of the factors involved. Ai is the effect of the ith level of one factor. Say A and Bj is the effect of the jth level of another factor, and say B and Eijk is an error effect of the jth level for another factor, and say B and EJK is the error effect which is assumed to be normally and independently distributed with zero mean and a constant variance. These assumptions regarding behavior of EJIK are necessarily for drawing inference by adopting known statistical methodology. The methodology that is adopted in the analysis of variance techniques, by which inference is drawn by applying f-test. For f-test, it is necessary that the observation by adopting various designs to be discussed subsequently, it have to be ensured that these assumptions are satisfied by the observations, otherwise no valid inference can be drawn from their analysis. Question here is, what is ANOVA? ANOVA is a statistical technique that assesses potential difference in a scale level dependent variables having two or more categories. The categories can be 3 or 4 or even more, but it should be more than 2. Researchers and students use ANOVA in many ways. The use of ANOVA depends upon the research design. Commonly, ANOVA are used in 3 ways. If the attribute is 1 or the variable is 1, we say it's a one-way ANOVA. If there are 2 variables used for this in this statistical technique for comparing 2 or more population, then we say it's a two-way ANOVA. But if the attributes of the variables used for comparing the populations are more, then we say it is n-way multivariate ANOVA, or also we say it is MANOVA. For example, an ANOVA can examine the potential difference in IQ scores by countries. Now, in this case, the IQ is only a one variable or a one attribute differentiating different countries. So, we say that it is a one-way ANOVA. The ANOVA table is a standard table used to summarize the analysis of variance, calculations and results. Let's see what is one-way ANOVA. Since this chapter is about the summary of analysis of variance, we will be summarizing various ANOVA techniques which have been described in the previous modules. So, in one-way ANOVA, it refers to the number of independent variables, not the number of categories in each variable. One-way ANOVA has just one independent variable. One-way analysis of variance is a statistical technique in which only one criteria or a variable or attribute is used to analyze the difference between more than 2 population groups. For example, difference in IQ can be assessed by a country. Countries can be 2 or 20 or more in this variable. Now, coming to two-way ANOVA, it is also called factorial ANOVA. Factorial ANOVA can be balanced. Balanced means have the same number of participants in each group, or it can also be unbalanced, having different number of participants in each group. It means that if we are comparing 3 groups of populations, then each population can have a different number. Two-way analysis of variance is a statistical technique in which 2 criteria or variable or attributes are used to analyze the difference between more than 2 population means. Next is the n-way ANOVA or MANOVA. A researcher can use many independent variables, and this is an n-way ANOVA. For example, potential difference in IQ scores can be examined by country, gender, age, group, ethnicity, etc. simultaneously. Since it is using many variables, we call it as MANOVA. Now, let's look at a general procedure of ANOVA or the ANOVA test. In an ANOVA, a researcher first sets up the null hypothesis. The null hypothesis assumes that there is no significant difference among the groups or the two or more populations. The alternative hypothesis assumes that there is a significant difference among the populations. Students must calculate the F ratio and the probability of F. Next, the student should compare the critical value P of the F ratio with the established value alpha. In general terms, if the P value associated with the F is smaller than the P is equal to 0.05, the null hypothesis will be rejected and the alternative hypothesis is accepted. Means that if the significance value is less than 0.05, then we can say there is a significant difference between the groups. It means that the alternative hypothesis is accepted, rejecting the null hypothesis. So, we can also conclude that the mean of the groups are not equal. What is F-test? The aim of analysis of variance is to see whether there exists any real difference between the population or the treatments, or they are only the errors of sampling. For that, we start with the null hypothesis that all the population or the treatments or the samples are equal as far as their effects on the yield of the crop or any other characteristics is concerned. Means the difference between these population is 0. If we suppose for a T for the sample or a population effect, the null hypothesis will be, or you can take mu also, this is just a notation, mu1 is equal to mu2 is equal to mu3 up to mu-n. Either this hypothesis will be disapproved or accepted within the limits of chance error. This can be tested by calculating the ratio between the sample variance and error variance and then testing its significance by comparing it with the expected value of the variance ratio at the desired probability level. This ratio between the two variances is expressed by the symbol F, or we call it as the F test or the F ratio. If V1 and V2 are the two variances based on the degrees of freedom respectively, then F is equal to V1 over V2. The observed value of F is compared with the value given in the table for V1 and V2 degrees of freedom at any desired probability level. The levels in which we are generally interested are 0.05, 0.01 and 0.001, or we can also say 5% level, 1% level or 0.1% level, and this is the level of significance. Let's take an example. The population variance or the mean square population variance is equal to 7.5 and the area variance or mean square error is 3.5. Then F is equal to, or F calculated is equal to 2.4 because it is the ratio of Vt over Ve or 7.5 over 3.5. The degrees of freedom V1 is equal to 4 and V2 is equal to 8. Now the value of F at 5% level of significance for V1 is equal to 4 and V2 is equal to 8 is 3.84. This is known as the critical value or the F table value. The observed value of F is less than the F table value and is not significant, proving thereby there is no significant difference among the sample or the populations. Had the observed value of F been proved significant, its interpretation would have been the differences among the population are real and not due to the errors of sampling. After that, you can see the summary table of one-way ANOVA, in which between sample treatments its sum of square is referred by SSTR and degrees of freedom by R minus 1 and mean squares by MSTR, which is a ratio of SSTR upon R minus 1. This has been explained in the previous chapters also, but since this chapter is a concluding or a summary of all the previous chapters or modules related to analysis of variance, this is just a repetition of what table we have seen in the previous modules. So next also you can see the summary table of a two-way ANOVA, and the only difference is that in two-way ANOVA we are taking two attributes or two variables for study, and in this the block treatment and for the F ratio for the samples or the population and F ratio for the blocks is being worked out with F for population is equal to MSTR upon MSE and F ratio for block is equal to MSR upon MSE. Next is the table of analysis of variance for complete randomized design, and in this you can see that F is equal to VT upon VE. Next is the summary table for the analysis of variance for a randomized block design, and in this randomized block design F for population is VB upon VE and for block it is VT upon VE. Let's take a question and try to understand that how this analysis of variance is worked out for a manufacturing company. A soap foam is filled in grams by 6 machines in 3 different shifts, and each machine fills 8 bottles in one shift. Now we want to test if the productivity of each machine varies in the 3 shifts or not. Here you can see the table. So in the columns you can see the various machines, and you can see 3 different tables which is showing the 3 different shifts data, and in the block A you can see the soap in grams filled in 8 bottles. So here you can see the table depicting the machines and the shifts. On the columns 1, 2, 3, 4, 5, 6, there are 6 machines, and on the left hand side you can see the shifts, and this is the summary of the bottles of machine 1, which is 400. Machine 1 fills 406 grams of soap in all the 3 shifts, and like that you can see the total number of volume of soap filled in the 3 shifts. When we work out the sum of squares of bottles, it comes out to be 3687.50, and like that you can find out the sum of squares for machines and sum of square due to shifts. Next is the ANOVA table shown, and it shows the F value at a alpha level of 1%, and you can see for machines it is 24 and for shifts it is 7. Like that you can make the assumptions and then based on these results you can interpret whether this assumptions is true. Which assumption? That the productivity of the machine in the 3 shifts, it varies or not. So the students, let's now summarize what we have learned in this module. In this module we have learned that each business or a social problem may have a different statistical testing design. A design for an experiment defines the size and the number of experimental units. The manner in which the treatments are allotted to the units, and also the appropriate type of grouping of testing units. We have also understood that in a statistical model, number of factors are involved with one or more terms representing error effects. The effects of any factor can be either fixed or random. Explanation and equations of one-way ANOVA, two-way ANOVA and MANOVA have also been mentioned in this module with a generic procedure to solve the ANOVA problem, and you have seen that how we have described the way of solving the ANOVA problem. Thank you.